very generous introduction. It's my great honor to speak in this symposium together with such this honored list of speakers. So it is my great pleasure to introduce our recent line of work about modeling single cell and spatial omics data. So since this is named for Ken, I want to say that when I was a graduate student at Berkeley, a teacher of a class, David Aldous, used the applied probability book from Len, and I was so impressed from the clear writing of the book. So I found this other book myself. So that's how I first got to know Ken before I came to UCLA as a faculty member and met him for the first time during my interview. So I want to say that the model, we hope it can account for this new technology and the data many people analyze, single cell data. It started with single cell RNA-seq data probably 10 years ago. And this is just a very typical figure you will see in those data generating or analysis papers, right? So each color means one cell type. And this UMAP plot is supposed to visualize cells in 2 dimensions. Every dot is one cell. And in this case, the cells are labeled with discrete cell type labels. And in some analysis, people will say, oh, the cells may follow a continuous trajectory. So this is another example. And this trajectory inference task, which means that you try to find an order of the cells based on the cell's gene expression data. And typically, the gene expression data vector is thousands of dimensions to start with. So how would you find the trajectory? And how would you assign a value, a scalar value for a cell along the trajectory? That is called a trajectory inference. And the value each cell receives is called a pseudo time value. That's the second example. And then people have the spatial transcriptomics technologies, which has become popular in recent 5 years. So in this case, you can record the location of each cell or a group of neighboring cells, depending on the resolution, spatial resolution of the technology in a tissue slice. So you have more information about these cells because you have the 2D spatial locations. And here we're just showing 3 genes. So if we want to summarize all these data into like a matrix form we're familiar with, the data matrix will have tens of thousands of cells, and the feature number is very high dimensional. If it's just RNA sequencing data, you will get like thousands to tens of thousands of genes. But if you have other types of omics, for example, the so-called ATAC-seq to measure the DNA open chromatin regions, then this number of features can be in the millions. So this is the data you want to analyze. But besides, we typically have some additional information about the cells. For example, biologists may have clustered the cells and annotate each cell cluster as a cell type. So you may have a cell type label, or you may have the cell pseudotime value I introduced, or you may have the cell's locations in spatial data. And sometimes optionally, you may have the batch information of the cells. They are not measured in the same batch, or you may have the condition information, disease versus normal. So these are the data you are going to analyze. What we want to do is to provide a statistical model. Why do we want to do this? First of all, I think as for all statistical model, we want to interpret real data. We hope that the model parameters carry interpretation. Specifically, they may reflect scientific hypothesis. So for example, given a cell's covariate, for example, cell type, if we want to know if we can infer the expected expression of a gene, then that is something meaningful, right? Whether the gene is a marker gene for the cell type or not. And for these parameters, we want to do statistical inference, right? Not just the point estimates, but also the uncertainty. Second, I think this is something useful for analysis is to generate alternatives to real data, because in many cases, we don't have the capacity to generate any conditions, right? Generate data from any conditions. So we have just data from a particular condition. But what if the data were generated in another condition? So the alternative hypothesis we have in mind, can we reflect that by modifying our model parameters? In fact, when we do power analysis based on a model, we are doing this. We're actually imagining alternative data with different sample sizes, and then what will be the power. So for this purpose, we should be able to do sampling from the model. So one thing people may criticize, especially scientists, is that our models are wrong, right? But we know that some models are useful. So therefore, I think model diagnostics is essential. How can we say whether a model is reasonable or not? So let's get back to data. So we have the following considerations when we model the data. To me, the first question to answer for any real data modeling problem is, what are the samples? So in this case, we consider cells to be samples because I think it satisfies our original intuition of sampling. When we measure a group of cells, we only measure the things that we capture, right? We don't measure everything. There are potentially other cells from this patient, but we don't measure. We miss during the experiment. So cells are samples, but the features like genes, we consider them to be well-defined quantities. So like we can measure the same gene in different cells, but if we just measure cells by two different people, we will not get the same cell. So we consider cells to be samples from some m-dimensional distribution of our interesting. And we consider the parameters that carry scientific meaning to be parameters of this m-dimensional distribution for the m features. How about the other two matrices? So we want to be conditioning on them. So using this vertical thing to mean conditioning. So that's the goal for our modeling. So in other words, for now, we will consider these cell states and the cell design to be given, to be fixed. Then condition on them, we want to model cells as samples from an m-dimensional distribution, conditioning on this. So this is a line of work started from 2019. And at that time, we just want to provide a tool for doing the power analysis for single cell data, like how many cells do we want to sequence, how to set the total sequencing depth. But when we started, we were always naive. In this case, we consider the M features to be independent, and we model the log transform count data. So these data are often counts if you're doing sequencing, right? Counts the sequencing reads. So we use the log transform data and we assume Gaussian distribution just to make things simple. But then we realized that this independence assumption is very unrealistic. So that gets us to version 2, where we consider the pairwise correlations of the M features. And in that case, we modeled cells in discrete types. So in other words, we fit one m-dimensional distribution for each cell type. Another change is that in this version, we directly model counts instead of log transform counts. So we use count distributions, such as negative binomial. But as I previously mentioned, right, the data can have more forms, such as in the continuous trajectory. So then in version 3, we allow cells covariates to be continuous as pseudotime values along a trajectory or the spatial locations. So that is the current version of the model. Furthermore, we go beyond the numerical data to generating sequencing reads. So this provides a foundation for us to generate sequencing reads in a synthetic experiment. So for my talk today, I will focus on this SC Design 3 model, but I want to give credits to my students, former student Vivian, who worked on the first version, Tianyi, who graduated, who worked on second version, and my current student Wang Ao, who worked on the read simulator. So about the current version, SC Design 3, which was done by my student Dongyuan and Qin Yang, who's in the audience. So we hope to have a model with interpretation. So let's focus on the right. With the model, we hope to provide some parameter estimates about a gene's expected expression in the cell type, the variance in the cell type, or the gene's expected expression in the trajectory along the pseudotime, or gene's expected expression in the two-dimensional space. So these are the mean functions we want to estimate for every gene. And also, we want the model to provide a likelihood, then we can use likelihood to do model selection. So we can compute the BIC, for example, to compare different models, which I'll show one result. And finally, for the alternative generation, we hope to modify the model parameters to create alternative models from which we can sample from to create alternative data. So we can make one gene as not differentially expressed between two cell types. The real data may have some difference, but we can wipe out the difference. Or we can generate cells in one cell type, even though the real cells may belong to more than one cell type. So that's about the model. But I think many people got to know about our work because it's a simulator. So actually we branded mostly as a simulator. So by simulator, we mean that we can do sampling from the data. But in my talk today, I will not talk too much about the simulation functionality. Rather, I will just use the simulated data via sampling from the fitted model as a way to show that the model fits reasonably well. Okay, then what is the model? For illustration, we will focus on one value, one random variable of cell I with feature J, right? So feature J in cell I call this Yij. To model its distribution, this is the Jth feature, I will take these information. One is cell I's state covariates. So for example, cell type, and also the design covariates, including batch and condition. So I will just use these two, for example. So what I hope to fit is such a conditional distribution jointly for yi1 to yin, the n features jointly given xi and zi. So specifically, using the notations, we have introduced the y as the data matrix, typically a count matrix, x as the state covariate. If it's cell type, then it's one-dimensional categorical variable. If it's pseudotime with p lineages, because those pseudotime inference algorithms can allow more than one trajectories, then it will be p continuous random variables, or spatial coordinates, then two-dimensional continuous random variables. And the batch and condition, both categorical. So first of all, we want to model the marginal distribution for each feature J given xi and zi, and we denote this distribution as fj. So in the most flexible form, we allow this distribution to have three parameters. Mean, the location parameter. This is the dispersion or variance parameter, sigma, and p is the zero inflation parameter. So we may not need all of them for some data, but we just write them here for the general form. So in the most general case, you would have a link function for each parameter and try to link the parameter to your cell state covariates xi, this vector here. And for the batch and condition. So here we just use the simple modeling approach by adding them as intercept effects. So specifically, to fit such a model for each feature J, there is a method, or there's a model, it's called GAM-LSS, Generalized Additive Model for Location, Scale, and Shape. So here, the F, G, and H are allowed to be nonlinear. That's why it's called generalized additive model instead of linear model. So the fact that we would allow it to be nonlinear is because the effect of the pseudotime or the spatial location may not have a linear effect on the gene mean expression, even on the log scale. All right, so this Fj is the distribution for feature J. These two things, xi, zi, state covariates, design covariates are being conditional on. And these are the functions that will be modeled as the additive model. So for the distribution itself, we have these options in the current implementation. So from Gaussian to Bernoulli-Poisson negative binomial and the zero inflated versions. And for the effects of the cell state on expected gene expression, we have these different considerations. So if it's a discrete cell type, then the effect is just like an intercept. If it's one lineage, we use one spline function. If it's p lineages, we have p spline functions, and spatial location, so far we use Gaussian process. But I want to say that these are still modifiable depending on how we want to model the mean expression of a gene along with the state covariates. And the joint distribution is where the pairwise gene correlations were considered. So suppose that we want to link the n marginal distributions of the n features into a joint distribution. How do we do this multivariate modeling? So we use the approach called copula, which means that you can use the copula function as the joint distribution of n uniform 0 to 1 random variables. And we use the most popular, I should say the simplest form of copula called Gaussian copula, which is actually defined by a multivariate Gaussian distribution. So to put it shortly, this copula function is defined based on an m-dimensional Gaussian distribution. And in other words, what we did is to convert every feature into a standard Gaussian distribution, and then we model the joint distribution of m standard Gaussian distributions. So what is needed here is this m by m correlation matrix, because every dimension is standard Gaussian, so 0 mean, variance 1. So what's needed is the correlation matrix. And we have a dimensionality issue, because as we have seen that the number of features may be bigger than the number of data points, samples, the number of cells. So how do we do this? Because when this m is bigger than n, we cannot really have a likelihood. So here we use a technique called Vine copula, which is a way to approximate Gaussian copula by imposing this structure on the m features correlations. So in other words, the first layer just connects some features, just connects the features into like a vine. And then the second layer is conditional on the nodes in the first layer. And the third layer is conditional on the third layer. So this construction will give you a correlation matrix that for which you can compute the likelihood. So this is basic from the likelihood computation purpose. And finally, how do we estimate the correlation matrix, right? We want to go for the Vine copula. What do we do? So we use the two-step approach in the current implementation. First of all, we estimate this conditional distribution CDF function for each feature separately. And then we just use those fitted marginal distributions to convert my jth feature in ith cell into the CDF value. So then we use the CDF values to fit the Gaussian copula. But one thing to note here is that there's a gap between my fitted distribution, if it's a count distribution, and the Gaussian copula, because Gaussian copula assumes continuous random variables, but the count distribution is discrete. So one thing we did here is to add this random random interpolation to convert the discrete CDF function into a continuous CDF function. That's what we did in the middle. And finally, once we have the model fitted, when we do sampling or so-called simulation, what we did is to sample synthetic cells, draw synthetic cells from the fitted model. We first start from this fitted m-dimensional copula, and then convert. So here, each variable is uniform between 0 to 1, and then use the fitted CDF function for each feature marginally to convert it back to the count value as just a quantile. So that's the fitting part. So for our students here, I hope this illustration will make things more straightforward. That is, I'm just using two features for illustration. Starting from real data, marginally, we fit a distribution. And the bottom is the joint modeling, for which we will use the marginal distribution to convert the data in each dimension into standard Gaussian value via the CDF and then inverse Gaussian CDF. And then we fitted a multivariate Gaussian distribution here. With this fitted, we can do sampling, and the sample values will be standard Gaussian in each dimension. Then we use the standard Gaussian CDF function to transform them to CDF values. Finally, we use the inverse of the CDF function of the distribution we fitted at the top to the original space. So that's how we get the synthetic data. And our hope is that if the model fits well, then the synthetic data will look like real data. So therefore, here we just want to show that when the real data has the pseudo-time values inferred by some other algorithm, we use that as the cell covariates. This is what we get. So the middle is our synthetic data sample from the fitted model. And in comparison, this is a method which is a simulator based on the deep learning using the GAN. So this is what this method gets. So one thing we want to say here is that this method doesn't use the pseudo-time information as we used. And the other thing is that somehow maybe because of the author's implementation, the running time is very long, much longer than ours. And one more thing we want to say is this quantity, which is commonly used in single cell field, called this MLISI, mean local inverse simplicity index, which I think is a very interesting quantity that may need further investigation. So this value takes range from 1 to 2. So 2 means that the two groups of cells mix perfectly. 1 means that they are completely separate. So you can roughly think as, okay, if I pick a cell and get a neighborhood, so if I pool these two batches, the test real cells and my synthetic cells, pool them together, do the UMAP, then this index measures, okay, in the neighborhood of a real cell, how much, what proportion is the real cells, what proportion is the synthetic cells. So if the two proportions are equal, then this number should be equal to 2. So it measures how well the two data sets mixed. And I think it's kind of an interesting way to do model diagnostics, or at least in practice, because we want to know how well do the synthetic cells mimic real cells. And this is about how we do on spatial data. So we basically fit a model using the spatial locations as a coordinates. So the top row is the test data we left out for validation, and the bottom is our synthetic data. And finally, this is about a technology where you measure protein and RNA jointly. So we want to show that we can preserve the correlation between protein and RNA in our synthetic data. So these are just to show that the model fits well to the data. And these are the parameters. So first thing we can get out of the model is we can get expected gene expression along cell state covariates. So essentially, when the cell state covariate is the pseudotime, we can get this mean curve, which can be interpreted as the smooth relationship of the gene given the pseudotime. So we think this can carry some biological meanings for data analysis. And this is the mean function, 2D function in the space. So we can use this as also a smooth version of the data to see where the gene is very highly expressed, lowly expressed. And moreover, as the Gaussian copula have the gene-gene pairwise correlations, we can report that here. So one thing you can see from it is that the two T cell subtypes, their correlation matrices are more similar to each other than with this monocyte cell type. And furthermore, this Vine copula provides this Vine structure as an approximation of the correlation matrix, which can somehow give us interesting information as which genes are the hub genes. These are more densely connected, or these have higher correlations with each other in the first layer. So that thing is also something we think can be useful. And furthermore, we can use the likelihood to compare models. So in this case, for example, if the cell covariate is pseudotime, what we can do is that we can apply 3 competing methods for inferring pseudotime values from the same data. So Monaco proposed this concept of pseudotime, and then we have method T-scan and slingshot. And so basically, each method will give you a different set of pseudotime values. We can input that into our model, and then we can compute the BIC. And then from this, we can show that, okay, the BIC values are reported here. And if we compare them, slingshot has the best value, and which is actually kind of consistent with the visualization and also people's empirical evidence. Okay, and finally, I want to quickly say that about the alternative generation. So since we can fit a model for one cell type, then if we force ourselves to assume, okay, there's only one cell type, we fit a model on these real cells, actually they have 2 types, then this is the synthetic null cells. So what we preserve is the per gene mean, per gene variance, and gene-gene correlations. But because the model assumes one cell type, and also ultimately because of the homogeneity in the underlying Gaussian distribution in the Gaussian copula, so the synthetic cells will look very homogeneous, and the gaps among real cells will be filled in. So we hope this can use as a negative control in some way for real data analysis. So one application we did is to consider this single cell strategy in every data analysis. People often do this, is to divide the cells based on clustering. And then for every gene, they want to compare the two clusters to say whether the gene is a differential expressed gene. So these two steps, clustering and differential expression, use the same data. And this issue is known as the double dipping. So what's the problem here is that even though the cells look like one homogeneous cluster, you can still force the cells to be divided. And then the genes used to drive the division will be found as differential expressed genes. So these genes may not be meaningful cell type markers, or they cannot be used to define cell types themselves. So to address this issue, we applied this synthetic null generation idea. That is, by assuming the real cells follow one null model of one cell type, we fit that model and then sample from that model to get a synthetic null data, which serves as a negative control just as in a regular experiment. And then for any analysis pipeline, people choose clustering algorithm, DE test, one test per gene to compare the two clusters. So here we want to be flexible because there are many options out there, and people commonly use the pipeline called SURAT for their analysis. So from the pipeline, what people will get out of it is a per gene DE score. For example, it could be negative log p-value from the test. So the DE score, the magnitude is going to reflect the likelihood that a gene is a DE gene. So we want to counteract this double dipping bias because you are confirming the data-driven bias using the same data. So we hope this negative control because we apply clustering and DE to it as well. So same pipeline, two data set. The null DE scores we got out of the synthetic null data, we think it can serve as a negative control. So the intuition is that if a gene has a target DE score and the null DE score of similar magnitude, then the gene is unlikely to be a meaningful cell type marker. So therefore, what we expect out of it is to do a difference. We call that contrast score. So target DE score minus null DE score. And if the null is a reasonable null data, then we hope that the majority of genes' contrast scores should be symmetric about 0. And only a small proportion of the genes will have large contrast scores. That means their target DE scores are much bigger than their null DE scores. And therefore, these genes are the interesting genes we want to find. So for the last step, we will use a previous approach we developed and which was adopted from the knockoff literature in statistics to do the p-value free FDR control. So basically, we will use the contrast scores to find the threshold instead of using p-values. So this is one example to show that the negative control idea is making some difference. So again, contrast score is the difference between target DE score and null DE score. And these two sets of genes are well known to be marker genes of CD14 monocytes or CD16 monocytes. And these are well known to be housekeeping genes. So what we want to say here is that the housekeeping genes cannot be meaningful cell type markers. And if you look at these genes in this plot, right? So if you look at target DE scores only, these will be the decision if we just run SURAT. SURAT will try to draw a threshold here to call differential expressed genes. But you see that some housekeeping genes are getting very small p-values. This is like negative 10 to the negative 30. However, on the null data, we see that housekeeping genes also have very big DE scores. So therefore, when we do a contrast, which is the y-axis, you see that the housekeeping genes' contrast scores become lower than those of the marker genes. So therefore, using the contrast scores, we can better distinguish these two gene sets than just using the DE analysis alone. And if we pick two genes, for example, one is the CD16, a well-known marker gene, and the one is this housekeeping gene. So what we want to say here is that using our method, the rank of the housekeeping gene is much lower than the rank of the marker gene. So here we're doing a box plot because we're calculating this across different differential tests. So we see that this is a general trend, but using SURAT, they are not so separable. And this is something people typically do in bioinformatics that is called gene set enrichment analysis. Again, taking each gene set,
we want to see where this gene set is enriched in the ranked DE gene list. So what this figure shows is that the marker genes are enriched in the top of the gene list returned by cluster DE, while housekeeping genes are enriched more in the bottom. So these are 5 differential expression tests in the top, in the column. And Surat, their differences are not as clear. And finally, another application showing that we can use the strategy to decide whether 2 ambiguous cell clusters should be merged. So this is from a Nature paper about the Drosophila Visual System Developmental Atlas. So these are single cells. And in the author's original annotation, they have defined hundreds of clusters, but these are the clusters that the authors couldn't give different names. So they were just given the same name, but like T1 cell type, but stay as clusters. So we want to show here that running our analysis for each pair of clusters, we found 0 DE genes, which indicate that merging is reasonable. But look at how many genes were returned by Surat. It's actually pretty big, can be as large as 2,000. So this is the preprint of this work, and we're still finalizing it. We hope to improve it based on the feedback we receive from the field. So about ongoing extensions of this approach, this model. So first of all, this synthetic null idea we have implemented on spatial data. So the idea is that what if you generate spatial data with no clear domain boundaries? Because people typically want to do this spatial clustering to define spatial domains. And for this task, there has been many methods already, like Bayes space is one method. So this is how it performs on real data, seems to agree pretty well with annotation. So if we generate synthetic null data to see how reliable each boundary is, this is what we get by running Bayes space on the synthetic null data. So you can clearly see that it's indeed much more ambiguous than the real data. And this was led by my visiting student, Siqi from China. And finally, my two model extensions led by my students. One is we hope to extend the model to this newer technology called Perturb-seq, where you can perturb genes in single cells by the CRISPR technology. So in this case, you will have additional information about what genes are perturbed in each cell. So we hope to extend the model to count for that. So this is Qingyang's preliminary results showing that we can have the synthetic data look similar to the real data in some sense. And also, can we generalize the model for population level single cell data? That is, the cells may belong to multiple individuals, and each individual can have its genotype, right? This is related to genetics. So how can we extend the model for this purpose? So this is our preliminary result. So we call this SC Design POP. And this is the only model simulator available so far. It's called SplatPop. So one thing we can show from the result is that in terms of mimicking real data, we are doing better. So this is, again, the MLISI value for similarity between synthetic cells sampled from the model and real cells. And this is led by my student in statistics, Chris, and also the first-year student in computational medicine, or I should say biomathematics program, Yihui. They're both in the audience. But I'll have some future work and open questions, especially for this expert audience about the optimization. So first of all, so far, the things I showed were only the point estimates of the parameters. So we haven't used the uncertainty quantification yet. And second, even though I said model diagnostics is very important, but so far, we only showed the similarity between sample synthetic cells and real cells on the UMAP space using that MLISI metric. That's it. But we didn't have anything more rigorous beyond that. And also, I think the biggest bottleneck we are encountering is the computational efficiency. How can we do things fast, right? Especially for ATAC-seq I mentioned, the feature number M can be in the millions. So for that, we are currently having a problem. How do we do it? Do we really need to model the original dimensions, or can we model some reduced dimension, just as Matthew's previous talk mentioned? Yeah, that's one possibility. Finally, I think this is something very interesting, but I haven't got a reasonable answer from biologists yet. That is, how do we interpret the gene correlations conditional on a cell state covariance? For example, if the cell state is cell type, we see that the cells are not like the genes are not independent. And that is an important part of our model to be realistic. But how do we interpret these correlations? What do they mean biologically? Why would there be correlations? Because in the single cell field, people can do both clustering analysis and trajectory analysis, as I mentioned. But in this case, you can easily fit a trajectory in this cell type. But is there really a direction, or it's just gene correlations that are not directional? I think that remains an open question. Or maybe possibly we cannot answer this question using data alone. So I think that is a big open question to the field. So finally, I want to give credits to all my students for the projects I mentioned, especially Dongyuan, who has been a major player in the third version of the model, and also the cluster DE work I talked about in the end, and also my other students for all the tremendous work they put into the extension and the speed up of the package. And I want to say that Dongyuan is currently looking for a job, and he will be a fantastic candidate. Thank you. Yes.
Yeah, Jessica, that was a fantastic talk. I'm very enthusiastic about what you're doing. And you mentioned about model diagnostics.
Yeah.
And I wanted to say, and it might be because I think you know, Chris.
Yeah.
I think you have the solution already, because if you have a method for generating a lot of data from your model, simulating like you do, and then you have real data, you can use machine learning. It's an idea from Jerry Friedman from 10 years ago, but we have it in our generative model paper survey, where you just use machine learning and you find out whether the labels can be found of the simulated compared to the ordinary.
Yeah.
And it's actually, Jerry was teaching, that idea is behind what the GAN. It's the idea between of this competition in some sense, where the computer plays against itself in trying to distinguish what it's simulated.
Right.
You know, and so you have this, it's the same idea. So you can just do the model diagnostic in that way.
Thank you.
So, but the other thing I have to add, which is about, I work a lot on biological meaning interpretation. And so you sort of have to be very pushy with the biologist to ask them to give you some cells which you are going to project onto your cells. Extreme cases or special cells where something is known, which you don't put in the analyst, but afterwards you put in the projections. And so sometimes you have these extra covariates that you didn't put in the analysis that made the human.
Yeah.
But to give the interpretation of the directionality of what's going on. Yeah. So that extra projection helps a lot with interpreting. And I always call that cheating, but I do that all the time. Yeah. That is, you're not telling me something, you know, tell me something more. Yeah. I can project it on.
Yeah, exactly.
Lovely talk.
Thank you so much. Yeah, thank you, Susan. Yeah, I have been talking with Chris already because we're trying to modularize this software. Yeah, thank you.